Oxford. She's best known for her surface. She's done a lot more, and today she's going to talk about analyzing cortical organization and the relation to cognition using machine learning. Thank you very much.
Everyone hear me now? Thanks for having me here today. So today I'd like to talk a bit about using machine learning to analyze cortical organization and start to investigate its relationship with cognition. So the cerebral cortex is a highly convoluted thin layer of grey matter at the outer surface of the brain, shown here in between the yellow lines in the image at the bottom. We know since the advent of cytoarchitectural mapping that it can be subdivided into a number of functionally specialized regions. We don't know exactly how many of these regions there are due to the complexity of the brain, but we know that they can be characterized by unique patterns of cellular organization and neural connectivity. And these connections allow coordinated behavior between distant regions of the brain, supporting higher order cognition. Interestingly, the human brain is 3 times larger than its nearest primate relative and much more convoluted. This is particularly true of the prefrontal cortex, which is an area of the brain responsible for higher order cognitive processing such as decision making and working memory. This region is also implicated in a number of neurological and particularly psychiatric diseases. The challenge is that the brain is extremely variable in terms of its shape and functional organization, particularly within the frontal lobe. So here I highlight variation in the low spatial frequency or core scale folding patterns in the brain. And here this highlights a particular complex of functional regions. The frontal eye fields, which is in bright blue, the posterior eye fields in pink, and the language region 55B. The topology of these regions is extremely variable from subject to subject. This presents significant challenges in terms of knowing how to compare from one location in one brain to a neuroanatomically equivalent location in another brain. Traditionally, what we use is image registration, so this is the process of smoothly deforming an image, brain image, until it looks more like another image. But this process of smooth or diffeomorphic transformation does not allow breaking of topology, which then presents a problem with this known high degree of variability in the cortex. Partly for these reasons, it's becoming accepted that the best way to analyze cortical features is to do it through cortically constrained processing. This is because representing the cortex as a sheet allows us to better capture the true geodesic distances between regions. So this has significant implications, for example, for image registration. So if we had a very small misregistration in the cortex that represented perhaps by a small shift causing 2 different banks of the sulcus to overlap across subjects, then you can imagine that 2 completely different functional regions will be compared as the same thing in downstream analysis. Whereas if we represent the true distances between these 2 regions, it's much less likely that this level of gross misalignment would happen. And surface-constrained surface-constrained smoothing prevents mixing of signal between the cortex and the subcortical white matter. This is particularly important for example, functional imaging data, because these are signals that originate from the cortex, and the signals from the white matter are assumed to be noise. So for these reasons, there are now a range of available pipelines and data sets for cortical surface analysis. So the first one was FreeSurfer. So generally, the way in which it works is you start with a volumetric image and use image processing to remove the skull. You perform labeling of regions, which requires a spatial normalization step of the volumes to a granular average space. intensity-based segmentation of the cortex, fitting of a tessellated mesh. It's then common practice to project this surface, which looks like this, to a sphere because it simplifies alignment. FreeSurfer is a tool that's developed for adults. It can be used for children over about 2 years old. It's been used in monkeys. We have used an adapted version of these pipelines for the Human Connecting Project, which is a very big open database that's acquired about 1,200 high spatial and temporal resolution functional imaging, microstructural imaging data of young healthy adults. I'm currently working on the Developing Human Connecting Project, where we're repeating these pipelines in babies. We've got a pipeline now working for the neonates, which are acquired from 29 weeks gestation to about 45, so it includes a large number of preterm babies. And we're working on fetal data. And the biobank will also have cortical surface processing at some stage soon. So as part of all this, I was, as I said, involved in surface registration for the HCP. I developed a tool called MSM. This is for multimodal alignment of brain morphology and function. It works using, it's a spherical alignment approach with a series of control point grids which are allow course-defined alignment of spheres, and is implemented with discrete optimization, which just means that rather than allowing each point on the mesh to move to any other location, it's constrained to move to a finite set of endpoints. This type of optimization is more robust to local minima and noise in the data, which is important because functional imaging data is quite noisy. It's been shown to significantly improve aerial correspondence, and by that we mean improvement of alignment of features that reflect the underlying functional organization of the brain, so functional imaging data. And this has led to a better mapping of cortical organization with the Human Connecting Project's multimodal cortical parcellation, which you show here. I'm going to talk about that in a few slides' time. By better representation of organization, which is closer to the underlying cytoarchitecture, it improves our interpretability of predictive models, but it's still, MSM ultimately is still a topologically constrained smooth registration algorithm, so this is still a limitation of the approach. So bearing this all in mind, cortical surface analysis can offer very significant benefits if you're interested in features of the cortex, but it is very challenging to get your data into a cortical surface representation. It's very dependent on good resolution of the cortex, and this is not necessarily the case for developing data sets and clinical data sets. This has required the development of specialized pipelines for babies, for example, where we additionally tune a step where we refine the fitting of the tessellated mesh based on intensity. This can be quite temperamental. It's very sensitive to acquisition properties and doesn't generalize very well. The full pipeline is very slow to run. So can machine learning help? Certainly. So we're already seeing big advantages for using machine learning, particularly deep learning, for image processing tasks, including image registration, image segmentation, and cortical parcellation, which essentially is a segmentation process. We're starting to see applications for cortical surface mesh extraction within the DHCP. This is what we're working on, and predictive modeling of phenotypes. These are available now and robustly show better performance than the state of the art. These are very challenging problems that we're working on. And in the rest of the talk, I'm going to give some examples of applications of these different methods. So just before I talk about these, I thought I'd just go through why convolutional neural networks and deep learning are potentially advantageous for these problems. So CNNs were based on a model of human visual system. Again, I've forgotten, oh no, the reference is here for this figure. So when we receive information, visual information, it's passed from the retina to the back of the brain, and the region V1 is essentially an edge filter detector. It detects high spatial frequencies in the visual field. Information is then passed hierarchically through regions which detect increasingly more complex textures before regions in the in the inferior temporal cortex are known. Individual cells are known to activate for individual objects in the image. Artificial neural networks are designed in the same to mimic this. So you learn a series of layers of artificial neurons. The artificial neurons are spatial filters. As you go through the network, you learn increasingly complex filters, starting with edge detectors and ending up with individual neurons will fire for individual objects in the image. And this is advantageous because it allows us to compare imaging data without performing spatial normalization, because this is learning a compressed representation of the images through their relationship with these spatial filters from which we can compare them. And this means that CNNs are now having state-of-the-art performance in certain subfields of image processing and computer vision, so no more than in image segmentation. This is understandable because, as we've already just said, CNNs are being trained to identify objects in the image, so naturally they have all the information that they need to also segment them. So this is just an example of a very popular network for originally proposed for segmentation. So here you pass in your image and some training labels, and from that you can make a prediction of the labels. That you can then perform segmentation on unseen data. So these types of networks are having a real impact on medical imaging now, and as we heard yesterday, are already being used in clinical practice. This is an example of one recent application by my PhD student Samuel Bird. So here we're performing segmentation, real-time segmentation of the fetal head from ultrasound. It does this using a UNet, but with an Bayesian implementation. So it uses something called Monte Carlo dropout to randomly drop weights during inference to generate a series of samples, and from this we can generate error bars on the segmentation in real time, and this can guide sonographers towards obtaining measurements from optimal ultrasound views and thus reducing error of measurement. And more to the point, colleague Martin Ratchell from my previous group in Biomedia at Imperial trained a network to perform tissue segmentation. So it was trained on the output of traditional methods, optimization-based methods based on expectation maximization. These methods are very dependent on certain image pre-processing steps, including bias correction and image registration. Image registration in particular can fail catastrophically sometimes if you have a large rotation of your, or a very large sort of affine translation of your images. Whereas he found that if you trained a deep network on the outputs, these were actually robust to these data sets and worked better. Machine learning is being already used for volumetric registration. So here, rather than estimating a pixel-wise segmentation of the data, it's estimating a displacement for each point in the image. This is an example by Adrian Delsa, the Voxelworth algorithm. So here it uses a variational generative model to estimate a velocity field, and then the novel, what they call squaring and scaling layers to estimate a diffeomorphism. We've not yet got an application for cortical surfaces, but we are working on it, so watch this space. Machine learning has had a real impact on our work in cortical parcellation. So this is the HCP parcellation again. So here, Matthew Glasser and David Van Essen from the University of Washington, University of St. Louis in WashU. They performed painstaking hand annotation of MSM-aligned group average multimodal imaging data. So this includes morphological features and functional features. Doing this, they are able to identify 83 regions previously reported by histological studies and determine 97 entirely new regions. The reason why there's a large number of new regions is because there's a large areas of the cortex that haven't been thoroughly charted with cytoarchitectonic studies. So this generated the following group average map, but as I've already said, we can't just propagate these to individuals through registration because the registration isn't reliable. So what we instead did was train a classifier. So here they used a method by Carl Hacker, and this is a MLP, so it's a neural network, but not a convolutional one. It doesn't take into account any spatial context. It classifies each pixel independently, and they trained 360 binary classifications, so that's 170 regions each hemisphere. And we're able to capture individual variability by only training each classifier on data where the individual subject's data agreed very closely with the group average. And this then generated these very individually tailored parcellations of the cortex for individual subjects, where all this data is registered. So you can see there's a high degree of individual variability left after image registration. Obviously, doing this as an MLP and training 360 different classifiers is perhaps suboptimal. It's slow to train all of these different methods and predict on them. And also, without taking into spatial context, you can't give any guarantee of spatial contiguity, and there is a 3% of examples in the data set where they're unable to detect the region at all in the individual. So here we retrained it as a UNet. So it's trained on the output of the MLP, and you can see with these dice scores that there's good agreement. It's very difficult, though, to know which is better because there is no underlying ground truth. Once we've got these regions, we're able to build macro-scale models of brain connectivity networks. So essentially, you can do this for functional or microstructural data. So we can average functional, this is fMRI, time series for each region, and then estimate partial or full correlation matrices, which is shown on the bottom here. Or we can use what is known as diffusion MRI to estimate structural connections. So this is approximating the neural pathways. These types of networks are historically analyzed with graph theory. So graph theory allows us to make inferences about local and global topological properties of the network. For example, investigating the tendency of the network to cluster into modules around densely connected hub regions, including, for example, the thalamus, which is a relay station between the body and the motor and sensory regions in the cortex, for example. So these are allow us to compare across populations and investigate or look into or make assertions about how different pathologies impact, for example, the efficiency or the resilience of networks. But machine learning has particular advantages. So for one, in principle, it allows us to make personalized predictions. So we are able to train on a set and then apply our model to some left-out data set to see how well it generalizes. So this allows us to make predictions for individuals, but given the extreme heterogeneity of the brain and the heterogeneity of many phenotypes that we're looking at related to cognition and behavior, psychiatric diseases, the fact that these diseases are, our understanding of them is evolving and our characterization and diagnostic tests are evolving, this is an extremely challenging problem. But what machine learning can also do is, if we use the right methods, return the features that were most important for making its prediction, and this can allow us to start to develop mechanistic models of these different phenotypes. So here we have an example of how we're able to perform machine learning-based prediction of age, and these connections show which structural connections in the brain are most impacted by healthy aging. And here we have the areas of the brain which are most impacted by preterm birth. We're also able, I also performed some analysis on the HCP, the adult data. So here we use random forest regression to predict fluid intelligence from the HCP features. So we've got 110 cortical image features that we used for annotation. So they included brain morphology, cortical myelination, task fMRI, and rest fMRI. And these 110 features were averaged for each of the 360 regions of the HCP parcellation, with some then random forest-based feature selection. We don't get an amazing prediction, but what we do get are features back from the prediction which overlap with a number of regions that we know are involved in higher order cognitive processing and decision making. So this is interesting. We don't have to perform supervised learning. So in this study by Steve Smith from FIMRIB in Oxford, He used canonical correlation to compare functional connectivity networks for 119 subjects with all of the HCP demographic and behavioral markers. This found one strong mode that associated fluid intelligence with strong functional connectivity across all of the brain, and particularly in the default mode network. Interestingly, though, recent results are suggesting that we need to interpret these types of analyses very carefully. So in a follow-up work by Janine Bischobosch from, again from FIMRIB, she shows that what appears actually, so what we have historically been interpreting as variations in functional connectivity is actually being driven by residual misalignment of the data. So it's actually a morphological features rather than functional features of the data. Perhaps arguably to really get at the functional impact in behavior and cognition, what we need to be looking at is dynamic functional connectivity. So not just looking at the average properties of, or the correlation of the full time series, but looking at how the relationships and the interactions between brain regions are really evolving over time. And then, given that all of these previous methods are very much dependent on image registration, perhaps arguably we need to start looking at methods that are slightly more independent of registration or misalignments. So there's starting to be a real development in the area of surface deep learning. There are different branches of this, but here we're talking about the development of designing specialized filters that can be fit to tessellated surface meshes. And then these can be basically input into what, any existing 2D or 3D CNN architecture to make predictions. So using this type of approach, my postdocs share made some predictions of using the developing data, predictions of gestational age at scan using morphological and myelinational features, and this showed extremely good predictions within the range of error of the measurement. It's not a very interesting problem in the sense that the differences in brain imaging data over this period are so stark that making a prediction of gestational age at scan is not a very challenging problem, but it does do a lot better than an ROI-based analysis. So if we perform a random parcellation of the data and run it again, then the error is about twice as big. So it shows interesting way forward for the future. And another interesting development is that we're now starting to see methods for which we can use to visualize what our networks are learning, such that they become slightly less of a black box. So here, this method called GradCam was used. It returns the features that were important for the prediction, and perhaps not unsurprisingly, it's highlighting cortical myelination and cortical folding across the brain, which are rapidly developing over this age group. But the idea is to hopefully move towards using these to predict outcome for these vulnerable babies in future. I didn't want to talk about geometric deep learning without highlighting an alternative application for it, which is that you have methods for deep learning now that are designed for data graphs, for example, social networks, and they are tailored for networks which have irregular spacing as opposed to our regularly spaced meshes that we were looking before. And then these can be used to compare functional connectivity networks. So here, Ira Katena, who was a former colleague at Biomedia and is now in Twitter, used a Siamese network to compare functional connectivity networks, and interestingly used it to compare and predict autism patients, and also look at gender classification in the biobank. So because of the challenges that we've got with functional connectivity analysis, there's still some ways to go with the precision of this type of method, but it again shows a a lot of potential opportunity for future. And just I wanted to finish by talking about generative learning. So we really want to we want to understand the mechanisms underpinning differences so that we can improve our image processing models and better understand different pathological conditions in the brain. So here, a colleague, Jonathan Mokhtar from King's, developed a tool for prediction of changes in brain tissue maturation and shape using a Gaussian process model. So here we took all of the DHCP data, registered it into a common space, and then used the Gaussian process to predict differences in intensity and the deformation maps, whilst controlling for gestational age at birth, gestational age at scan, and gender. And through this, we're able to create visualizations of mean trajectories of brain growth, as well as if you fix the deformations, then you can look at changes in intensity and tissue maturation. Doing this, we're able to investigate the impact of prematurity on the brain. So here, the model is looking at what a brain would look like at term for different degrees of prematurity. And it's doing a really good job allowing us to start. It shows a huge amount of potential, but we still have some issues in the cortex due to the challenges of volumetric registration. So the plan is to go in the future to the surface and potentially look at deep generative models. So in summary, I got really quite fast. So obviously, I'm going to say, if you're interested in the cortex, use cortical surface constrained processing, preferably my software. But we have to be aware of making promises of personalized medicine. It would be really great if some point in my career I can predict individual markers of IQ and stuff, but we're a long way from doing that. To do related to the heterogeneities in the data and the phenotypes. But hopefully in future, interpretable AI can play a role in improving understanding of these underlying mechanisms. I just wanted to highlight that we're hiring. So we should soon have PhD positions available through our CDT here, and one of them would be relating to this generative model project that we were just talking about. And as part of a collaboration with the Primrose Centre at Oxford and Donders, we have a big project trying to improve the precision of personalised predictions from imaging data, and particularly we'll be working on deep learning surface to volume registrations. If anyone's looking for a postdoc, please pass them my way. Just lots of acknowledgements here to my team, but also the broader collaborations that I've been working on as part of the Developing Human Connectome project, the Human Connectome project, many collaborators to the slides here. And at the end of the slide here, anybody who's interested in using the data set, this is the links to where you can get the data and the processing pipelines from. Okay, thank you.
Thank you, Emma. That was an excellent talk covering a lot of ground. I'm sure it touches on a lot of areas that some of you are working on. And we have some time for questions. So if someone, yeah, shall I?
That's what fMRI does, yeah. Yes. I mean, there are lots of challenges with fMRI. I'm starting to look into fMRI EEG, which then gives you you've got both high spatial temporal resolution and higher spatial resolution fMRI. MEG, it also works for these pipelines, and PET as well. But anything you're interested in, if you're interested in the cortex, then my feeling, the evidence is suggesting that you'll get more sensitive comparisons if you go to a surface-based analysis. PET, no. So all of this is generated from fMRI. I mean, the 360 region parcellation is very fMRI focused.
So you mentioned that about the challenges in predictive modeling, right? So can you give some examples of what do you refer to by predictive modeling? Did they refer to like diagnosis, for example, the autism example you mentioned? Does that fall into your category of predictive?
Yes, so any phenotype. So I mean, what we like to do with the DHCP, our babies get invited back at 18 months for a barrage of cognitive tests. We'd like to be able to predict which babies, from their imaging data at birth, are most at risk of long-term neurological impairment. But this is extremely difficult because when we ask 18-month-year-olds to perform cognitive tests, they're not always cooperative. The data is extremely noisy. The characterizations of the questions that there are, we don't fully understand the underlying phenotypes, so it's extremely difficult label data to predict on. So whereas we can do extremely strong predictions of gestational age at birth, gestation age at scan, if we're trying to predict outcomes for these babies, it's just a much higher level. And this also applies, I don't have lots of experience with it myself yet, but if your goal was to predict autism from this data, it will also be much more challenging.
I think I've understood you correctly to say that registration is an issue for you. Have you looked at multi-scale registration techniques?
So MSM is multi-scale, course-defined alignment. I think the problem is more that you have these topology breaking transformations, regions are you would have to break the topology of the mesh. And I'm in the process of trying to assess how much of a problem that particular issue is, but even without that, there's such a degree of variability in morphology, and our estimation of fMRI maps, initial estimation of fMRI maps that we use to drive registration are dependent on the initial cortical folding-based alignment. It's all tied in, so image registration is generally a big problem.
I've been working on wavelet-based multi-scale methods which are very good for elastic deformations.
I was looking at, sorry.
Yeah, sorry, you carry on.
I mean, I was looking into an approach related to that for deep learning-driven alignment, which was inspired by that type of analysis. So I think it definitely has a place, yeah.
I'll be happy to talk.
Yeah, definitely.
We've time for one more question.
For attention disorder, because there's a variation of visual and audio depending on the frequency, have you looked at it? Because from certain colors and things like that, therefore you can see the vortex. This is a self-division for colors, each color or...
Sorry, could you repeat the beginning of that again? I missed it.
For attention disorder
Yes.
issues on audio and visual, there's a variation obviously on the frequency and colors of the spectrum. Have you looked at see if there's any variation on modeling when you look at it?
So the problem that, well, it's not a problem. So I've been working very much on healthy data sets at the moment. The DHCP, although we're collecting preterms, we don't have as many as we would like, and so our cohort is mostly healthy. And the HCP is all healthy, and so I haven't had the opportunity to work on those types of problems, and would be interested in doing so. I hope so. I think what we've really got at the moment is trying to predict cognitive markers from healthy babies, given the challenges in measuring them, is just really the hardest problem we could go for. So I'd be very interested in things where people know that are expecting a difference in the data or in the behavioural tests. That would be really good.
All right, let's thank Emma again. And I'm sure she's